right, good morning. I really appreciate the opportunity to be with you here this morning. And as Dr. Taubman said, I'm the chairman of the Department of Surgery, but I'm actually not going to talk to you about surgery because it's hard to follow and act like Ghazi, right? That guy is. I've used his models, and they're absolutely unbelievable. It's like you're in the operating room with the patient. So it's a huge service to all of us to have access to those kind of models for simulation. Really remarkable guy. But I'm going to actually talk to you about cancer. And in particular, we're going to talk about using the immune system to fight cancer. And that's something that's been in the news recently. I think you've probably all heard about this renewed enthusiasm for immune therapy for cancer. And it's actually an old idea. And people don't realize that. But this actually started in the 1800s with William Coley, who was a physician who actually noticed that his cancer patients, if they developed infections, they actually did better. They lived longer. And so he had this theory that by harnessing the immune system and causing inflammation, you can actually kill tumor cells. The body can kill tumor cells itself. And that was the whole theory behind Coley's toxin. And so he really came to the point where he would inject his cancer patients with infections to try to get them infected to fight cancer. And so he did that in the 1890s, and then it was kind of forgotten. Nobody really followed up on that for probably the next 70 years or so. And in the 1950s, there was a group of scientists, of immunologists by the name of Thomas and Barnett, who came up with this theory that we all develop cancer all the time, and our immune system surveys our body so that you develop a cancer and your immune system gets rid of it, and the clinical manifestation of cancer is just failure of the immune system to eradicate it. So that was a really interesting concept, and there's truth to it. I mean, there's some truth to that we probably all develop cancers that our immune system eradicates. And so if you think of cancer as failure of immunity, it's a really different way of thinking about cancer. And then in the 1960s, there was a clinical trial in bladder cancer where physicians actually used tuberculosis, so attenuated tuberculosis, injected it into the bladder of patients with superficial bladder cancer, and showed that they could actually cure patients with bladder cancer by using this technique. And that treatment still exists to this day. So this is not a new idea, but it's one that has gained a lot of traction recently as we really start to understand the biology of the immune response to cancer. And so we're going to go on for the talk here. I have to actually leave the room because I have a flight at 11:45, so I'm not sure I'm going to have time for questions at the end, but if you have questions that I can't say to answer, any of the Meliora folks or advancement folks can give me my email address. I'm happy to hear from any of you and answer questions. I'm heading actually to a scientific advisory board meeting for a company that we're working with on the West Coast to develop some of these immune modulation trials for cancer. And interestingly, the meeting is in Napa Valley. So I got a telephone call on Wednesday saying the meeting is not going to be in Napa Valley because the hotel is on fire. And the meeting has to go on. We have to have this meeting, but we're looking for an alternative place to have it. And I said, well, Rochester, we have wineries. I can guarantee you there won't be wildfires, and I'm pretty certain there won't be earthquakes, so why don't you come to Rochester? And it's a little hard to find 200 hotel rooms in Rochester at the last minute on Meliora Weekend, so that idea didn't work. So I have to go to, and I have an 11:45 flight, so we're cutting it close. So one of the reasons why there's a lot of enthusiasm is really related to Jimmy Carter's illness. Jimmy Carter's in his 90s, right? And you all heard in 2015 that he had liver metastasis and brain metastasis. So when you hear that from melanoma, most people think, okay, we're going to be hearing about Jimmy Carter's funeral preparations in 3 months. And now it's almost 2.5 years later, and he's actually doing very well. And last I heard, on his last set of images, there was no evidence of residual melanoma despite having widespread, essentially, metastasis from melanoma. So that caught a lot of people's attention and said, well, maybe there is something to this immune therapy for cancer. So what is immune therapy for cancer? And the problem is that people talk of it as it's one thing, and it's actually not. It's a multi-pronged attack, and there's many ways to think about it. The immune system is extremely complicated, and there are several things that have been brought to the therapy. Adoptive cell transfer is actually just taking cells from the patient's own body and usually taking them from the tumor, because the idea being, well, the ones in the tumor might be the ones fighting the cancer. Can we take them out of the tumor, grow them, and then give them back to the patient? And then you can take those T cells and you can genetically engineer them and really supercharge them and say, okay, let's just take these T cells that are in the tumor, rev them up, and give them back to the patient. And so those were kind of the early ideas. Immune checkpoint modulators, and that's the kind of drug that Jimmy Carter is taking. And that's really every immune response has a gas pedal and has brakes, right? So if you have an infection, you want a really robust immune response to tackle that infection, but when the infection is controlled, you want to dampen that immune response, you want to shut it off. And the immune system has many different mechanisms to shut itself off or to suppress inflammation, and one of those is on T cells. And so there's a molecule that pops up on T cells when you want to dampen an immune response, and it allows them to stop. So it stops. Think of it like sticking a cinder block under the brake so you can't put the brakes on. And then there's antibodies. So we've all, the antibodies are different. They're made by B cells. They float through the blood, and they can attach on cell surface proteins, and then they're flagged as bad things that you want to get rid of. There's cancer vaccines, and we've even come to the point where we can take a patient's cancer, we can sequence it and look at the genetic abnormalities and then design a vaccine particular to that patient. So the concept of personalized vaccines is something that's actually gained traction, and there's instances in certain diseases where that has worked. And then there's these overall immune system modulators that work on that, that balance between activation and deactivation, stepping on the gas and putting on the brakes. And we've worked in that. A lot of the work in immune therapies for cancer has really focused on the T cell. And the T cell is important because that's the final common killer cell in many instances, but this background of multiple cells in the microenvironment that tips that balance one way or another are important. And we've spent a lot of time trying to figure out how to optimize them. And so this is a picture, and this is a older paper in 2006, but I show you a picture of this guy who's really the father of immune therapy. He's been a mentor and a role model of mine for many years. He's a surgeon. He's the chief of surgery at the National Cancer Institute, the surgery branch in Bethesda. He's the only guy I know who went from being a chief resident to being a chief of surgery. He was hired as a chief of surgery at the NIH right out of residency, and he's been doing that job for 40 years. This is a patient of his that he treated with adoptive transfer. So he had this kind of epiphany when he was a resident because he saw a patient, and it was a patient with melanoma that had widespread liver metastasis like this, and then he saw the patient 6 months later and saw another scan, and the metastasis had disappeared. And he said, wow, what did you give this patient to have this incredible remission of the cancer? It turns out the patient wasn't treated. So this patient had a spontaneous remission of his cancer based on the fact that his immune system, for whatever reason, recognized it and started to destroy it. So when he saw that, he said, so he saw that, and he's been working on this for 40 years since seeing that. And this is a patient where, back 10, 20, 15, 20 years ago, we were isolating tumors, taking them out, isolating the T cells, take them into lab, grow them in huge numbers, and then just give them back. The idea being if there's some in there that are working, more is better, and more is better, and more is better. So it was just growing these cells, the bulk of them, giving them back to the patient. And in certain patients, we saw dramatic responses like this where all the tumors melted away. The problem is that was very rare for that to happen. You'd see it happen, but it happened in 1 out of 100 patients. And so the real question is why? What is the biology that's making this work in some patients and not in others? And then there was also some concerns about this because if you can take the immune system and really rev it on and really kind of pedal to the metal, put your foot on that gas and get it to go, can you actually cause damage if you can't shut it off? There's a reason why there's this balance. And what if you can't shut it off and then the patient starts having all kinds of problems from inflammation that won't stop? Can you take a system that's dormant and activate it? And can there be unintended consequences of that? So people started to get concerned about side effects in autoimmunity. So this picture is one where it made me think about taking something that's dormant. What are the unintended consequences of taking something that's dormant and activating it? And this is actually a real picture of my son Benjamin, not photoshopped. It's an actual picture of him in Chiang Mai in Thailand, where they obviously don't have any personal injury lawyers. It's kind of made me think of the whole concept of autoimmunity. If we grab the tiger by the tail, are we going to ultimately cause the patient problems? So it's something that we think about as we develop immune therapies. Are there going to be off-target consequences? Now, we started off by telling you I'm a surgeon, and I'm not talking about surgery. Well, why is that? I take care of patients with pancreas cancer, and this is a survival curve for patients with pancreas cancer. And this is actually an old slide from back in the time when I was a fellow at Memorial Sloan Kettering. So I took care of many of these patients, and this is actual survival where you have 10, 15, 20-year follow-up on these patients. And you can see that basically after 5 years, we have very few survivors, and at 10 years, there are none. And this is pancreas cancer patients that got resected. So if you know about pancreas cancer, the vast majority of patients at the time they're diagnosed, it's already spread. They never see a surgeon. This is the good ones. These are the ones that had resectable, localized disease and had an operation with curative intent. And we often, as surgeons, congratulate ourselves, well, that, we do these great operations, and they're complex operations, and we get the patient through it with little morbidity and get them home. And isn't it wonderful how successful we are at surgery? But time after time after time, despite the fact I did the best I could with the operation, the patient would come back, sometimes 6 months, sometimes a year later with recurrence. And I had this, it dawned on me that surgery is not the answer to this disease. Surgery is not solving it, so we need surgery plus something else. And that is what kind of led me down this path of investigating ways to use the immune system to augment our current treatments to pancreas cancer. Now, when you take a pancreas cancer out, this is what it looks like under the microscope. And you can see that these are the cancer cells here, the bright pink ones, but around it you have all of these other cells. And we just asked ourselves this very simple question. And sometimes 80% of the tumor is not cancer cells. Only 10 or 20% of the tumor is tumor cells, and the rest of the cells in the tumor are not cancer cells. They're something else. And so we asked ourselves, well, first of all, what are these cells? And then second of all, is this one of the reasons why this is such a bad cancer, that these cells are there supporting the tumor, helping it grow, helping it spread, helping it metastasize? And so we spent a lot of time just isolating these cells and cataloging them and finding out what they are. And it turns out most of them are immune cells, and they're a mix of immune cells. There are T cells in there, there are B cells in there, there are macrophages. So multiple immune cells make up this, what we call the stroma, around the cancer in the tumor itself. And this is just a cartoon depicting that. The blue are the cancer cells. This is a blood vessel. You have all these immune cells. Here's a macrophage, here's a T cell, here's a neutrophil. And all of these things can conspire to kind of cloak the tumor in the shield of armor so that the immune response can't work. And one of the things that we notice when you look at these cells and actually categorize them, the vast majority of them are these cells that are called macrophages, which are pretty primitive immune cells. And these macrophages sit there, and they do provide that cloak. There's no T cell that's going to work because these macrophages are there just spilling out these chemicals that will prevent a T cell from working. And so when you talk about the immune system, you have to talk about two different phases of the immune system, the innate immunity versus adaptive immunity. And think of it this way. The innate system is really a very primitive system. So plants have an innate immune system that has macrophages. Lower organisms have it. And only when you get up to jawed vertebrates do you get this very sophisticated adaptive immune response with T cells and B cells that are very specific for antigens. So it's almost like this magic laser bullet when you get over here. You know exactly what the infection is, what the antigen is, and these cells can multiply and they can attack it, whereas the innate is more just if there's an infection, there's signals that go out and you get an influx of cells that try to fight the infection. So it's just this level of sophistication in the adaptive immune response that is potentially important. And so what happens is, and this is just comparing the innate to adaptive, the timing, innate's going to happen very quickly. Those cells, as soon as there's sense that there's a pathogen or an infection there, the innate cells will arrive. The adaptive is later. This is with these more primitive cells, the neutrophils, macrophages, and dendritic cells, whereas the specific T cells. And the difference is that they have receptors on them that can be rearranged. So there's millions of rearrangements of those receptors that can be very specific for a certain type of antigen that can be recognized by the immune system. Whereas the recognition here is just it'll recognize patterns from bacteria, but they're not specific. It's pretty non-specific. And I'm going to show you some movies of that will help you understand that. And then, as I said, in terms of evolution, this is in much lower organisms where we only see this adaptive response in higher vertebrates. And this is what an electron micrograph looks like of a T cell that we talked about attacking a tumor cell. And I said I was going to show you some movies, and this is, I think one of the wonderful things about being at the University of Rochester is these movies come from a colleague and collaborator of mine, Min-Soo Kim, who some of you, I think he spoke at this a couple of years ago, so some of you may have heard him talk. But we have some of the best basic immunologists and immune imagers that allow us to really image the immune system in vivo like this, so we can measure it live, and so we can take this from our mouse models of cancer as well as imaging the human immune system. So when you pair together a guy like that, an amazing basic immunologist, scientist that has these incredible tools for imaging, with me, a clinician who designs clinical trials and does translational research, we work together, and it's these clinician-scientist pairings that really advance the disease. A lot of times the basic biologist will be working on some mechanism related to the cell biology, but making that leap of how that's going to help a patient and how we're going to take those laboratory discoveries to clinical trials and to patients. And so here at the University of Rochester, we have a great group of basic biologists and immunologists that I can work with to take this to patients. And so this is just I'm going to have to go back here, I think, to run the movie. But this is an example of that innate immune system. These are neutrophils, so those are the more primitive immune cells. And this is just, actually, this is a thermal injury. So this is a mouse ear where you create a thermal injury, and then you just see these neutrophils, they just traffic, and they come, and they just go on. So they're there to deal with the fact that there's inflammation there, and they've got to come, and they've got to deal with any infection that could result from this. So being able to image this in real time like this is an extremely powerful tool. And if you go to the next one, this is actually a yeast infection. Those long rod-like things are yeast. And you can see these cells coming in. And if you watch where that arrow just pointed, see that cell just gets into that hyphae and kills it. So that's dead. You can see it happen over here. It gets into that. And so we can image this at this level that we could have only dreamed about really even just a few years ago to try to figure out the biology of exactly what's going on. What are the cells we want there? What are the cells that we don't want? And then this is one looking at T cells. So those other ones I showed you was more innate immunity, so pretty nonspecific. This is a T cell that is just targeted exactly to an antigen expressed on that cancer cell. And the nice thing about this, if you watch this movie, we can actually see the cancer cells dying. As they die, they turn a yellowish red color. So you can see the T cell. It's recognizing its receptor on the cancer. You can see that. It's almost like engulfing the tumor. And as it goes on, notice that's going to change color and turn red. And so we know that, and you can almost see the cell dying. You can see the membrane breaking up. Another one here where the T cell is encountering the tumor. It's kind of feeling for it. It's going to meet that receptor on the tumor, and then the change in color where it turns yellow or red indicates cell death. So we can image these killer T cells killing tumor cells in real time, which is just amazing. So immune regulation, again, gas pedal and brakes. The immune system's designed to sense the danger signal, and then trigger a robust and a coordinated defense against it, and then refine that response from a generalized one to a specific one. And that's what we think is going on in cancer. And then it wants to dampen the response or shut off the response when it's no longer needed. And our point in cancer is that's probably happening too quickly. It's still needed, but the cancer somehow outsmarts the immune system to shut that response down. And so I kind of had this epiphany one day. As a surgeon, we deal with this all the time with wound healing, right? So we make incisions in people and they have to heal. So what happens? Wound healing is a very well choreographed physiologic response. So what happens first? Platelets come into the wound, right? Because you've got to stop the bleeding. What happens next? Neutrophils come into the wound because you've got to fight infection. What happens next? Macrophages come into the wound because you have to continue that fight against infection, but you also have to start thinking about remodeling and healing that wound and going from scar tissue to more physiologic tissue. So wound healing, this normal physiologic response, has to happen. You need this inflammation, then you've got to shut it off when the wound is healed. And it's our theory that cancer actually tricks the immune system, and it's almost like perceived as this chronic wound that still needs to be healed. And then if we could trick it into thinking it's a chronic wound that needs to be healed, we'd be better off, but it actually tricks it into this dampening. We've got to dampen it. The inflammation's been going on too long. And so there's a lot of analogies between normal wound healing and what's happening with the immune response. So how do we subvert this normal mechanism to shut off the immune response? We don't want to shut it off. We want to keep it going. We want to stick that cinder block under the brake if those cells are fighting the cancer. And you can look at both sides. We've actually focused on this innate side with these immune suppressive macrophages. I told you those come there. They prevent a T cell from working, and I'm showing you those great movies showing if a T cell could get there, it could kill the cancer cell. So we've focused on that. A lot of people have focused more on the adaptive, on the T cells, making these kind of supercharged T cells that can overcome T cell exhaustion when the T cell is starting to think, well, we need to be shutting this down. And we can genetically engineer these, and I'll show you a couple of pictures of this. This is something that's been in the news recently, a CAR T cell. So that stands for chimeric antigen receptor. We just did a trial here in patients with leukemia that resulted in this therapy becoming approved by the FDA. And the bigger trial is about to open, and we're one of the few centers in the country where this therapy will be available. But this is an antibody that's usually made by a B cell, but you can genetically engineer it to stick it on a T cell. So if that antibody recognizes this on the surface of a lymphoma cell, it has the signaling intact in the T cell to turn that T cell on and turn it into a killer T cell. So you're taking part of a B cell, part of a T cell, putting them together and making this magic bullet. We can also engineer T cell receptors, which is a more common way that T cells work, where the antigen here is presented on a receptor and the T cell recognizes it. And so we can take a T cell out of a patient, engineer it, and then grow up lots of them and give them back to the patient. And then these checkpoint inhibitors, what I told you that Jimmy Carter is on, is this whole system here where you've got an antigen in the tumor. The tumor's presenting the antigen saying the T cell, I don't belong here, there's a problem, the cell should be destroyed. But then you have this upregulation of this protein called PD-L1, and that's the brakes. A T cell that the receptor encounters PD-L1 is going to shut off and say, okay, it's time to shut off the immune response, because that is what happens when, after a robust immune response, when you want to shut it down, you upregulate PD-L1, and that shuts down the T cells. Well, let's block that with a drug. Let's stick that in there. Let's stick that cinder block under the brake and not shut that off and keep it revved up. So that's what the checkpoint inhibitors are. That's the drug that's had such success in Jimmy Carter's melanoma. We've been interested in the messaging that goes on between the tumor and the bone marrow to get these cells to come, because they all start in the bone marrow, and the tumor has this communication going on with the bone marrow to bring these cells to the tumor microenvironment where they can come and suppress an immune response. And this is just some work that we've done with this protein. You see this little red dot here? That's a chemical called CCL2, and that's a little message that the tumor is sending out saying, okay, it's time to shut off the immune response. When it's time to shut it off, that gets upregulated. It attracts these macrophages from the bone marrow. They come into the tumor and shut off the immune response. So the tumor is actually communicating with the bone marrow saying, shut off the immune response. And we've discovered that these signals are coming from the tumor, and we have a drug that can block that we've used now in pancreas cancer. We just completed a trial with that, showing that we've dramatically improved response rates to chemotherapy in patients that get this immune modulation by shutting that message down. So very interesting, this dynamic discussion going on between the tumor and the bone marrow attracting these cells. And this is just an example of a patient from this, and you can see that the T cells that are the killer cells are red, and they virtually aren't there before treatment, but when we block that message, you can see they start to come in and they start to infiltrate. And so those are the helpful killer T cells that you want activated. And I've gotten rid of the suppressive ones, so now they can work. So this is what I started with. What is cancer immunotherapy? It's a lot of things. And there's people working on all of these different angles in terms of attacking the immune system, really figuring out that balance between immune activation and immune suppression. And when you think back to the experiments that Steve Rosenberg did, and I actually did too as a resident working in the lab, is we used to just take these T cells out of tumors, we'd take them to the lab, we'd grow them up, and we'd give them back to the patient thinking if some is good, more is better. The problem with that approach is we were probably giving back suppressive T cells. We didn't know what we were giving back. We were just giving back whatever we grew out of the tumor. As we get to this level of sophistication of understanding the exact biology of the tumor microenvironment, we can really manipulate it so that our foot is on the pedal and the line to the brake is cut so that we can keep this immune response to the cancer going. So what are the pressing questions that we still have to answer? Well, one is how do we combine these with standard chemotherapies and radiation? That's an interesting question, because if you think about standard chemotherapy, what does it do? It's pretty nonspecific. It acts on killing rapidly divided cells. The whole concept behind chemotherapy is that cells that are rapidly dividing are cancer cells typically, and so just give chemotherapy. And that's why you have side effects from chemotherapy, right? People lose their hair, people get bad GI toxicities because those are cells that are rapidly dividing that also get destroyed by the chemotherapy. So I'm telling you, I just turned on this adaptive immune response against cancer. Don't go give chemotherapy that's going to wipe it out, right? So we don't know how to time that, or whether we should just be giving the immune therapies alone and forget about the chemotherapy or do it sequentially. So we're doing a lot of work to figure that out. And then how do we prevent the collateral damage? How do we, when we take the tiger by the tail? And how do we know which patients are most likely to respond? This is some really interesting work that we're doing now. We've got these incredibly sophisticated instruments where we can monitor the human immune response. We can look at 30 different parameters of a single immune cell and be able to tell if the macrophages are activated or are they suppressing, or the T cells working. And we can do that, and it's my hypothesis that we can take a biopsy from a patient after just a couple of weeks of treatment and say, yes, this is going to work for you, versus no, that's not going to work for you. And that's really important because if you look at chemotherapy for pancreas cancer, only about 20-25% of patients respond. So we give 100 patients therapy for 6 months, we only help 20 or 25, and at the end of that we can't tell you why. So having this kind of customized ability to figure out if you've turned on an adaptive immune response, the likelihood that that's going to work is much higher, and so we would continue the patient on that, and if it's not working, then switch them rapidly to somebody else. So this is all work that's ongoing. I've got a huge team of people, of both clinicians, translational scientists, basic scientists working on this. It's a wonderful environment here at the University of Rochester because we have so much strength in all of those areas that we can actually take interesting findings from the laboratory right to the patients. And that is my goal as a pancreas cancer physician. My theory is that every patient with pancreas cancer should be on an innovative clinical trial because it's been 30 years with very little progress in the improvement in the treatments of that disease, and unless we're doing that, we're not serving our patients. So I've got 3 minutes for questions. I do have my assistant waiting at the thing, and she has a lead foot, so I think we'll be okay. We'll get to the airport on time. Yeah.
So, getting actually to your first question about the ongoing work, have you considered looking towards nanoparticle therapy or nanomicelles and small vesicles and targeting with various other targeting molecules like hyaluronic acid to sort of combine these nanotechnologies to form something that will actually use EPR effect to target tumor cells more specifically versus a systemic response and toxicity with safety therapy?
Yeah, so there's two questions in there. The hyaluronic acid is an interesting one because that, if you look at that the picture I showed you, that all those cells in there, the chemotherapy can't get to the cancer cells because the stromal cells are so packed in there that it's a question of getting it there. And so hyaluronic acid is one of the reasons why in there. So there are therapies in pancreas cancer right now, when you test the HA levels in patients, and if it's high, then you put them on this drug that can kind of bust up the stroma to get the chemotherapy in. Your other question was about nanoparticles, and that is another way to deliver things. And we've actually taken a different approach to this, where the drugs that we're using are actually working in the bone marrow, preventing this whole orchestration of cells from happening. But we do have some really interesting data that's kind of hot off the press we're working on right now in the lab. Scott Gerber, who's a research scientist in my lab, has recently discovered using this nanoparticle technique that if you put a specific cytokine, IL-12, into the tumors, that you have this, you really have this profound immune response, which to the point where we were like curing mice with pancreas cancer like we had never seen before. And I said, Scott, that looks fantastic. Like, let's wait for the tumors to grow out because we need to take those tumors and find out what's going on in the microenvironment, isolate those cells. And he said, Dr. Linehan, I'd love to give you tumors, but I cured all the mice. So, so the good news is if you're a mouse with pancreas cancer, we got you covered. So we got to now take that to humans. So, but the question is what's happening there, and I think that interleukin-12 is driving the differentiation of these macrophages to the point where they're actually helping an immune response, not shutting one off. Yes?
Why do you think hyperbaric oxygen and or negative suction is beneficial for wound healing?
Well, so a lot of the physiology of wound healing obviously depends on oxygen delivery to cells. And so when you have this inflammatory response with all this fibrosis, it's a matter of delivering enough oxygen to the wound to get it to heal. And so with hyperbaric, it's basically like supercharging the hemoglobin so it can deliver oxygen to the place where it needs it most, often in a place where blood vessels are disrupted and you've got all these macrophages causing fibrosis. And so it's really a question of oxygen delivery to a healing wound.
And negative suction, which work was done by Walter Porres here
Yeah.
in 1962 or something.
Yeah, and that's actually revolutionized the way we deal with complex wounds, because we've just found that with these suction devices, they're really vacuum devices where you just accelerate wound healing. It's probably related to the exudate that comes out of wounds by removing that. It may relate to the oxygen delivery. If you're removing the exudate by suction, there's more oxygen making it to the wound. So that's been a big advance in complex surgical wounds. It's important. All right, thank you very much. I wish I could stay longer.